On Thursday then, we will take those extracts and someone in the molecular area upstairs will run what we call pyrosequencing. So are you all familiar with pyrosequencing as a method of sequencing? Do you know the difference between it and standard traditional sequencing? Maybe not. Do you want to take this one? Or do you want me to?
If you can take that.
So you know how when you do a PCR or sequencing, you add primers, well, for PCR you add primers and you add nucleotides? And you do your PCR procedure, which may take 3 or 4 hours. This is going to be really basic. 3 or 4 hours, if you will, and you get a result in the end. Yes, it matched or not on the sequencing. All those nucleotides matched. You put it in a database, and it says, yep, this is this organism. Pyrosequencing, first of all, the pyro part of it means that the way in which it's measured is by little poofs of light that go off as the reaction is occurring, as the hybridization, if you will, is occurring. But the method of pyrosequencing is different from other methods of sequencing in that as a nucleotide is added and it combines with the nucleotide from the target organism, makes a match here, if you will, and starts to develop more of it, if you will, that lights up and you can actually see that fact that it was positive for A, T, C, or G, depending upon what the concomitant nucleotide is. So you always put your nucleotides in, or the machine does, in the same order all the time. So as it's being synthesized, as that layer of DNA is matching up with the nucleotides that are being added in, it actually shows up as a light band, if you will, and as it's being synthesized, you can actually start to read the banding pattern. So you actually know before it's completed what the organism is theoretically. Now we let it run and we get the answer in the end and we do it in the end, but it's called a sequencing by synthesis. As it's being formed, you actually do get your results as compared to getting it all at the end. That's one of the differences, and the fact that there's light emitted, and that's the way, it's like the firefly light. That's the way this particular method works, if you will. So that's being done on Thursday. It takes about 3 hours, 5 hours?
Probably about 3 hours.
About 3 hours for the sequencing to be done. And then we get a whole bunch of pieces of paper at the end of that. And that's what Mary has in front of her. So on Thursdays, generally what we do is Mary runs through every organism that was sequenced. And it's usually somewhere between 10 and 25. Just depend on the week, the raw, and how many mycobacterium we have. Because we are a reference lab, we get isolates of mycobacterium from around Cleveland and also Akron. People just know every Thursday, Cleveland Clinic, pyrosequences their mycobacteria, get it into them on Tuesday or the very latest Wednesday morning early, and you'll get it in that run for Thursday. And literally people around the whole city know that. Did I make the run? Did I get there in time, kind of thing. And then Mary's group and Mary will go ahead and get it extracted and then get the pyrosequencing done. So we get together, and I'd like the residents to come at least once, we get together and we talk about what organisms were matched, what organisms do we come up with answers, and then if something doesn't match or work, we have alternative methods that we'll talk about and what Mary will do about it. So that's what this is all about.
Okay.
So now I'll let you take it.
Okay. Do you want me to show like?
Yeah, if you wouldn't mind.
Okay. This is a report off of after we make the extract. The first thing they do is they make a master mix and they amplify the product. And they amplify it. They just picked the rotor gene. They could have picked the light cycler. They could have picked. I can't think of what else they could have picked.
Smart cycler.
The smart cycler, but they just picked the rotor gene because nothing was probably on it at the time.
Yeah, and rotogene, light cycler, smart cycler, these are all methods of real-time PCR. And so that we just happen to use this format.
And you can use it to identify stuff, but we don't use it here. All we use it is to amplify. But what comes off is you'll get these graphs, and all you want to do is make sure that your patients amplify, and they usually amplify before 25, and this is the water. The water will eventually amplify no matter how sterile the water we buy. It'll eventually amplify, but it should be way later, and it came up around 30.
So you've probably seen graphs like this before. I'm assuming PCR graphs like this before. I know you have because it's been in your PBMs. But at any rate, yeah.
And then they also do a melt curve.
You understand what a melt curve is after you've hybridized in the process of making your amplified products, if you will. Then you separate it out and you determine how long it takes to separate out and at what temperature it separates out, and that's a melt curve.
Yeah, and this is sort of it's not a real specific melt curve. All you want to do is you want to see, if you have a peak out here, you know you've got a
Something's
problem.
wrong, something happened.
Yeah.
So this tells Mary and the group on Wednesday that we can run these, this
Yeah.
extract is fine.
Yeah.
Now, does somebody literally go and check that out?
It comes off because you get all these pieces of paper
Yeah.
and you're pulling the little amplicons off the machine.
Right.
So you're sort of looking at
Okay.
this, and
I mean, if you didn't get that peak, would you stop and not run it?
you'd run it anyway just to see what happened.
Okay.
But if it didn't work, you wouldn't wonder
Right.
why.
Why, and then you'd know why, absolutely.
And then this is what comes off the pyrosequencer. And these are the, you can, down at the bottom, this is the enzyme, and this is the substrate, and then you won't see those anymore. And then it'll drop it in a pattern, CTGA. And,
So those nucleotides are being added.
we'll add, and if they match, there's a blip of light. There's a few tricks. A is always a bigger peak than the other nucleotides, for reasons that I don't know. If you get peaks like this high, this means they're multiples. And you sort of judge by, this is a G, and this is probably 2 Gs.
Mhm.
And
It's not like absolutely quantitative, but you kind of visualize it and go, okay, if this is 1, this must be 2.
Yes.
It's basically what you do. And for some, you can actually get to 3.
And at the bottom, you'll see how the machine read it. Sometimes we'll go back and we'll check and we'll disagree with the machine, and that's called a manual read. And then the machine will give you the quality, and basically it's telling you whether they thought it was good. If we get a mixed sample, you will probably just get a series of peaks. Because
So you kind
everything
of know when you
matched.
look at, even my eyes, and I don't understand this as well as Mary does, when you get just tons and tons of peaks, there's something wrong in there. And you all know that you can amplify mixed cultures and get yes or no's about whether or not there was a specific organism in there.
Mhm.
But when you sequence mixed cultures, you get garbage. Sorry to
Yeah.
say that word, but it's true. Except for some new sophisticated sequencing methods that I'm hearing about, where you can actually look at stool with billions of organisms that are in there and be able to identify individual organisms by sequencing. It's fascinating. I don't understand it, but it can happen. But this can't. So you'll get, if you get, Mary will say to us, I think this is mixed, because you'll just have too many peaks.
And then this is the printout for somebody with really good eyes, and it's the same thing. It's just a basic summary.
A summary of what went on.
And I like to scan through here to see that the quality is good.
So you have some markers right off the bat about whether you really think this one worked or not. You look to see if there's too many peaks. You look to see if it starts out with the right letters. You look to see there was a melt curve. You look to see that indeed it passed, or the machine thought that it passed. And then, if you would talk about the database that we created.
Okay, we created a database. Once again, Marion
Yeah. Dr. Prokop.
Dr. Prokop created a database for TB and aerobic actinomycetes, and we can add to this database if we have, if something is not in our database, we can go to NCBI, is it NCBI?
I think so. The
Okay.
gene bank.
The gene
The
bank,
national gene bank.
and blast it. And if we get a good quality blast, we cannot report it out because it's not in our database, but we can send it to somewhere and have it sequenced. And when we get 3 of them that sequence the same way, we can put it in the database. And we have done that with, I believe, lentiflava
Mhm.
and Namoens. So we can add to this database.
So I think we've been doing this now for at least, oh gosh, I bet it's about 7 years now. What do you think?
Oh, yeah, because
I mean,
this
it's a
one
long
paper is
time.
from 1996.
Yeah, so what we did initially in the lab was, just as an aside, I don't know, you need to film it, but you can, there's no problem. The Pyrosequencing company at the time came to Dr. Prokop and said, we have this really cool tool that we didn't even know about at the time, what would you think about it? And I forgot to mention the other thing that's different about pyrosequencing, and I know Mary would get to it, is that it actually analyzes 30 nucleotide pairs, whereas most sequencers you hear about 500, 1,000, all nucleotide pairs in the genome study that's being done on humans, right? This just analyzes and gives results based on 30 nucleotide pairs. So the original thought was this is never going to work. Well, it does, actually,
It does.
for what we need to do with our organisms, et cetera. So Mary supplied, and the AFB lab, hundreds of mycobacterial organisms that we had categorized biochemically or phenotypically. And we also got isolates from Mayo Clinic, from Texas, from all over the place. And they had either identified them by phenotyping or had actually done some PCR work on it. They were in the game a little bit earlier than us, and so we knew what these isolates were. And for an organism to get into our database, it would have to have been very well characterized with good peaks and good bands on the pyrosequencing, and then it made it in the database. And we have lots of organisms in that database. But we continue to see others that don't fit in that database, which is what Mary was suggesting, and we don't put one in our database until we absolutely know that's what it is. So we expect 3 different patients to have that same organism that gives us the same answer in the gene bank, and then we put it in the database. But we don't just put it in because this time we did it and it came out to be XYZ. We really want to prove that it is, to make the database, a new word for me anyway, robust, meaning that when you get an answer there, you really can believe in it. And so that's how we decided to make that database. And we kind of abide by that, and we have all kind of rules about what organisms can go into the database, if you will.
Mm-hmm. And we have databases for mycobacteria, and we also have databases for nocardia.
Yeah.
And for the nocardia, we only do 20 base pairs.
Right, 20 base pairs. It's fascinating how this thing works. Cool.
Okay. So now we have what came out. We do a processing control, which is an MAI that we run at the beginning of the run. So it travels along just like the patients. So you extract it at the same time with the same UDO buffer and everything. And then at the end of the run, you have a positive control, which was a processing control that worked like last week or the week before. And then we have, I think they're using depth seawater, which is very pure water, as our negative control.
And sometimes we do get something in the negative control,
Oh, yeah.
but hopefully it's not mycobacterium something, some other organism that's probably not going to interfere at all, so we don't worry too much about it.
So then they developed this nifty little computer program that's called Identifier. And it takes the sequences that the pyrosequencing gives us, and it runs them through our database. And then it gives us the answers, and it gives us a score. And if it's not 100%, you don't take it.
We don't accept less than 100%.
Because if you look at our database, Mycobacterium tuberculosis complex is like 1 base pair off from an organism called Mycobacterium salata. And so if you're not 100%,
it doesn't get called.
No, it doesn't get called.